So you have to learn this cycle. Okay. So galactose can be converted to galactitol. Galactitol. So in the presence of aldose reductase. So again galactose can be converted to galactose 1-phosphate in the presence of glucokinase. All these cycles you just memorize better. And suppose if I think there is one clinical condition, galactosemia, isn't it? So galactokinase, classical galactosemia. So what is deficient here? So this whole cycle we have to learn better for galactose, that is for sure. Once you learn, we will see the clinical conditions. Oh, this is different. So deficiency of enzyme galactose 1-phosphate uridyl transferase. Suppose if this enzyme is deficient, galactose 1-phosphate cannot be converted to glucose 1-phosphate. Hence, galactose 1-phosphate will accumulate in blood, isn't it? Same thing will get excreted in urine. So that is, it will lead to galactosemia, galactosuria. Increase in blood is emia, increase in urine is galactosuria. Okay. So galactitol. So that means suppose if this is not present, galactokinase. So what happens? Galactose will remain in the blood. It can get converted to galactitol, which is very dangerous, and it may lead to cataract. Okay. So galactitol can get accumulated in liver, nervous tissue, lens, kidneys, and impair their functions. So like that, accumulation of galactose in liver results in depletion of inorganic phosphate for other metabolic functions. What are the clinical symptoms of galactosemia? Loss of weight, hepatosplenomegaly, jaundice, mental retardation, cataract, all these things. Okay. So that is one answer, beta, which we didn't discuss. What we have to check the levels of these parameters. So we have to check for the bilirubin. If bilirubin levels are increased, what does it mean? Heme catabolism is impaired. That means more number of RBCs are being broken down. Okay. And in case of liver disorders, we will find all these enzymes are increased, like alanine transaminase, aspartate transaminase, gamma glutamyl transpeptidase. These enzyme levels are increased. And in case of liver is not functioning, we know liver is the site for the synthesis of plasma proteins, albumin, globulin, prothrombin, fibrinogen. Hence, if liver is not working, albumin levels will fall down in blood, prothrombin levels will increase in blood. So urea levels, ammonia levels will also rise because liver is the site for the urea cycle, isn't it? So lipid metabolism, yeah, like fatty liver, I told you, no. So cholesterol is not being transported outside. Hence cholesterol levels will increase, triglyceride levels will increase. We know first pass of metabolism of drugs is in liver and second pass metabolism is in kidney, because drugs are not being like detoxified. You will see the half life of drugs, that means drugs will remain in blood for longer time and it is very dangerous to our body. Okay. Then increase bile acids, so bile acid content in the blood also will increase better. Okay. So all these things we will see if the liver is deranged. Apart from that, we are supposed to write about this jaundice. This is very very important. Vandenberg's reaction. Should I repeat better? Twice I explained you. If you want still, I will repeat, Niharika.
No, ma'am, it's okay for you.
It's fine for everyone, beta. The other students also. Please write if anybody wants. I don't mind. Heme synthesis we did. Heme synthesis is also a cycle only. Severe bowel cramps and diarrhea after feeding milk. Oh, probable diagnosis. What is this condition, beta? 11th question, check it. Tell me the diagnosis. Newborn baby. Okay. Now he is having distension, severe bowel cramps. That means lactose is not being digested in the body, no? This condition is lactose intolerance, isn't it? What is the deficient enzyme beta in lactose intolerance? Milk is not digested here. Check the enzyme beta.
Yes, ma'am.
Is it lactate dehydrogenase? Three hormones of adrenal glands. Daily I'm telling. And share that enzyme in the chat box, please, Niharika. Creatinine clearance test, we know.
Yes.
Three antioxidants. What are the three antioxidants? Vitamin A, vitamin C, vitamin E. Glutathione is also antioxidant. They did, what do they do? They just inactivate like the free radicals. H2O2, O2 minus, SO2, like that. Allosteric regulation also we did. High energy compounds. Energy if it is more than 7, then it is called as high.
Lactase, ma'am. Lactose
Okay,
intolerance.
okay. Thank you, beta. Lactase. Okay. Ritika, thank you. That's what I was confused. I was not sure. Okay. Then biomedical importance of carbohydrates. Biomedical importance in the sense commercially we use carbohydrates for treating certain diseases and all. That is the biomedical importance. I will see. Dietary fiber we did. So glycemic index, amphipathic lipids we will see. Biomedical importance of carbohydrates. Let me check. So we have studied glycosaminoglycans, no beta? So I'm sharing one slide, just see once, all of you. I also try to share it here. So what are we finding? This is the biomedical importance of carbohydrate. So certain carbohydrate derivatives are used as drugs like cardiac glycosides, antibiotics. Okay. Do you have anything to share, Ritika? You can share your screen by taking a photograph better. Is there any subtopic under our first chapter? Biomedical importance. Glycosaminoglycans you can write better. So much of importance is there, right? Under glycosaminoglycans. Using, I'll tell you. It's not strict. Hyaluronic acid, chondroitin sulfate, dermatan sulfate, isn't it? All those are biomedically important carbohydrates only. Mm, they are used to treat so many diseases, isn't it? This one. You can write this for the question what is given, beta. Biomedical importance. Biomedical, see here. Hyaluronic acid. We can use this for wound healing, as a lubricant, as a shock absorber. You can write this table only. And chondroitin sulfate, it helps to maintain the structure and shape of, that means this we can use if there is any fracture or something. Heparin can be used an anticoagulant. Dermaton sulfate also can be used, like this can be used commercially, though these are all natural substances, but still commercially also we can use these things. Okay, of course separate table is not given, but it is understood better. Okay. Amphipathic lipids. Phospholipids, another name is amphipathic lipids beta. Make a note, we did phospholipids, isn't it? So the most, see here, abundant membrane lipids are phospholipids, which are amphipathic in nature. So I'm using this screenshot to share with you. It's a twisted way of asking phospholipids question, amphipathic lipids. Okay, I'm sharing on the screen also. So I hope all of you have seen the answer. So what is amphipathic lipid? So all of the lipid molecules in cell membrane are amphipathic, also called as amphiphilic. Philic means they have hydrophilic, water-loving or polar end, and hydrophobic or non-polar end. Most abundant membrane lipids are phospholipids. They have a polar head group and two hydrophobic carbon tails. Okay, so that is nothing but you have to talk about phospholipids better. Okay. Glycemic index. What is glycemic index? It is given in our textbook. Okay, that means glycemic index is how fast or how slow they can release glucose. Okay, but still we will see in our textbook because it is there. So let me check the page number. Let's look. I'm not sure. Yes. So what is glycemic index? So area under the blood glucose curve after ingestion of test meal divided by area under the curve after ingestion of glucose multiplied with 100. So glycemic index, that means their definition. See, variations in the increase and fall of blood glucose levels after ingestion of different carbohydrate containing foods. The quantitative difference are assayed by glycemic index, which measures the time course of post. Okay, so what does this mean? See here, you are eating some carbohydrate, like you can eat rice, you can eat wheat, you can eat a banana, which are rich sources of carbohydrates you are taking, and you are just checking. Suppose this blue curve is indicating which has low glycemic index, like suppose it is potato, low glycemic index. So after taking potato, you will see after 30 minutes, the blood glucose levels have become somewhere at around 100. So after 90 minutes, gradually the blood glucose levels are falling down. That means that particular food item has comparatively low glycemic index. Whereas suppose if you take pure rice carbohydrate, we know it has rich glycemic index. That means once you eat rice, after half an hour, you see the blood glucose levels are rising up to 140. That means the food which can give more glucose in the blood after consuming it, this is called as a substance with high glycemic index. Whereas a substance which has more fiber and less carbohydrate, it will be releasing glucose very slowly in the blood. So such food substances fall under the category of low glycemic index. Now here few examples are also given. Glucose, direct glucose, what you take as glucose powder, it gives 100%. Okay, full glycemic index. Carrots 90 to 90, they also are good only. So okay, honey, bread, rice, banana, potato, like lowest one is soya beans, above which we have fructose, ice cream, milk. Milk has more protein and less carbohydrate. No, that's why they fall under anything which is below 50. They are the foods of low glycemic index. And foods of low glycemic index are good for diabetic people. Okay. So oranges, apples, ice creams, milk, fructose, soya beans, and all. Okay. So it is all about this question paper. Do you want me to discuss anything else better? You can write in chat box or we will stop here. Choose like good paper tomorrow and give me some alternative questions also. Tomorrow's timings, I will let you know better because tomorrow I'm going to Siddipet. By the time I come back, I don't know what time. After I come back, I'll go. So should I take any other question? Please let me know.